Yes, no, anything?
You're audible, Samal.
Cool. So we will be starting in few minutes. Yeah. So Dharya, let me know when you're live. Or should we wait for few minutes? So my camera is not working. So you have to bear it with me. I'm sorry for the inconvenience. It's cost. Yeah. Maybe I'll try with my mobile camera. Okay, cool. We are live. So I guess we can start our event. So on the behalf of so very on the first opportunity, I would like to say that I really welcome you all for taking out time and for joining in this workshop. I hope you will don't feel bad that you don't you. I hope that you will get a lot of things to learn about here. And meanwhile, people will be joining soon as of now. So what we are? So the SciComm Synapse is a community or an initiative focused on science communication that is helping scientists, students, and the creatives to learn how to effectively communicate scientific ideas to the diverse audiences using engaging formats like art, media, storytelling, and outreach. So here in SciComm Synapse, in the umbrella of SciComm Synapse, we have come with our project known as Kala Tattva. So what is Kala Tattva? Kala Tattva is our project where we maintain present science through sci art, and herein our main aim is to express is to provide a platform where one can express their love for science, the colors and imagination. Excuse me. So we conduct lectures, workshops, and interactive sessions with help of science artists in this Kala Tattva project. So why Kala and Tattva is important? That we have to discuss first. Then we will be going dive deep into the workshop. So Kala and Tattva. So Kala means what? Creativity. So when you apply creativity to science, it leads to imagination. People are joining. So I guess into the I have to turn off my camera because of the connectivity issue. Yeah, I hope I'm audible, audience. Yes, yeah, no. Something. You please open your mic. You can speak up on. One, two, three, four. Anything? Am I audible or not?
Yes.
Yeah, you can directly speak. Great. Okay. So before, so this is our basic introduction of Kalata. So why we have come up through this, and when we see that in presentation or scientific presentation, how do we represent our experiments and all? That is very important. Very important. So we have seen that students lack in that. So in this workshop, we will be showing you that how do Biorender works, and since it's a basic Biorender workshop, so here we'll be walking through how Biorender works, how you can access to it, and what all you can do it. So before I go move forward, I would like to invite Kirti to give you a basic introduction, theoretical introduction of biorender work, biorender. So Kirti, the stage is yours. Can you please go ahead?
Hi, thank you, Soumilya. Thank you so much. Should I open my camera or
Yeah,
just go with
you
the
can.
presentation? Okay, just
Actually, my
wait.
camera is not working, so
Okay.
last time it gave me dhoka.
Am I visible? Can you guys see me?
Yes.
Okay. So I'll be sharing my screen now. Just tell me if you see something, okay? Hello.
Yes, you are audible.
No, can you see my screen? Is my screen visible?
No, we can see your screen. Yes.
Okay, fine. So, hello everyone, I'm Kriti and I'm going to give you a small snippet on what Biorender actually is. So, Biorender as a whole is a scientific illustration tool. And I'm sorry, let me just put it in presentation mode. Yeah. So, Biorender is a scientific illustration tool as a whole. And it is a web based collaborative design platform which is specifically created for scientists and researchers to create quality scientific figures that can be utilized in publications. And one of the good thing about Biorender is that you don't need to download anything for Biorender. It is totally cloud based and maintained entirely within web browsers without requiring any software downloads. So then the question here arises: how is it different than other platforms? Because a lot of us as early career researchers or students, we don't know what to utilize when we want to convey scientific diagrams. So a lot of the time we use PowerPoint or Inkscape and things like that. So Biorender is different in that aspect as it is a tool specifically built for biological concepts versus general purpose driven design software. And it has strict consistency standard because it uses a unified visual language that maintain consistent representation across publications. So you can pick any paper that has a diagram or a figure by Biorender and you can easily replicate that in your own template when you want to make that. And specifically it does not require any artistic skill, so you don't need to draw anything on the template. You can just pick and drop whatever you want on the template. And by that, what I mean is, I'll explain in the next slide. So When it comes to the features of Biorender, it is a content library and a lot of templates, specifically 40,000 plus scientifically accurate icons that is designed by certified science illustrators across 30 plus life science field. So no scientific field is left alone, that oh, we don't have a representation of, for example, a rodent or a cell or a different type of animal model that a particular scientist might be using. And it has 5,000 plus customizable template for research, context, and publication. It has a specialized drawing tools and biospecific drawing tools for creating membranes, cell layers, and DNA structures in second. And nowadays, certain updates are also available in Biorender. So it has advanced features such as AI-powered tools, including timeline generator and cell-to-cell interaction generator. It has customizable poster builder for conference presentation, and so right. So what I meant by the earlier slide was that, see this slide here, this is the final slide, and this here shows the different sort of templates that you can find in Biorender. The first one here, which is written as the cancer cell part. So this is a whole package of cancer cells. Similarly, there are packages of say pathways or neuroscience or immunology. So these packages had certain cell types or pathways or molecules which represent those particular cell field. So such as in this cancer cell package, you can see you can either take a individual cancer cell or a metastasizing cancer cell as seen in this particular figure. Similarly, if you want to show a cancer cell which is, for example, heterogeneous, has different types of cell, or cancer cell that has been infiltrated by immune cells, so you can show all of this in Biorender. So in place of Biorender, if you were to use, for example, PowerPoint, then you have to make a circle, draw a circle inside it, then you have to draw arrows and so to differentiate cancer cell from different type of cells. So that's a lot of hassle. So Biorender basically eliminates all of those things and just makes it very simple for you to convey whatever you want to do. Similarly, for people working in plant sciences, here there's another template in Biorender which shows different types of root system. So this is, for example, a taproot system or a fibrous root system. If you were to replicate that in say Canva or PowerPoint, then you have to take an image of plant from the Google, then you have to try and draw roots from it with the help of lines and stuff using that. So again, that is a lot of hassle, it takes a lot of time, and in most of the cases it's not accurate. So another example of template here is, this is a template of brain, and this is what we call as a customizable template. So herein you can show whatever you want to in case of brain or neuron or synaptic information. So herein you can either, if you want to show neurotransmission, then you can put some cells from the Biorender cell template or program, and you can just put cells here, arrows here. Or if you want to draw pathways, and again there is a template or a program for pathways, and you can put that in here as well. If you want to show a synapse, then you can use different type of cells interaction. So all in all, it's just a very easy platform to uh, convey scientific information. So now, whatever I told you guys, Somil will be demonstrating you all about the different packages, the different programs that Biorender is having. So I pass my mic to Somil again. Thank you.
Thank you, Kirti. Yeah, it's really great to have you. So you have given really great insight. So before we go forward with the hands-on section, I would like to ask the audience, do you have any doubts or questions as of now? The things which we covered till now.
Yeah, if you have any questions regarding the presentation that I just showed, or the packages, or what I talked about, you can just directly ask me.
Yeah, you can ask. See, if you guys don't interact, then we will stop this workshop. Simple. Bhumika, any questions or anything?
Questions.
Guys, speak up. So,
Hi, Kritik.
yeah, Niharika, yes, you're saying.
Can we take this illustrations directly to publication with the free web tool, or do we need subscription for that?
Yes, we do publish with Biorender workshop. Biorender, you can create images and upload in publication as well.
Okay.
Yeah,
We
so
don't need
Biorender has
minimum one.
yeah, it has a premium package as well as non-premium one. So a lot of images and a lot of templates are also present in the non-premium ones. But again, if you want to have something very specific, for example, if you want to show some pathway that you are trying to, elucidate in the paper, then you have to buy some of the premium templates, not all, but some.
But this whatever we make using free web tool, they are publishable, right?
Yeah, everything is publishable. Biorender has this very cool thing that you can just make whatever you want to explain in the template, and you can directly translate or transfer it to your publication and paper.
Okay.
And it is one of the most, I think, I'd say, acceptable tool when it comes to scientific illustrations in research publications.
Okay. Thank you.
So, any more questions, or should we move forward with the hands-on session? What do you guys say? Audience, speak up.
I have a small doubt for Kirtan. Like most times when we do the free version, there is a limitation in the number of objects or templates we use, and we are not able to like.
उसके लिए तो भैया प्रीमियम वर्जन खरीदना पड़ेगा. Else, some universities or institutes they provide premium version.
Yeah, Sommel is right. Yeah, you won't have everything in the non-premium version, but if you, like I said to, I think Niharika's answer, if you want to show something that is very exclusive, and so you have to buy the premium version. And yes, I would like add on to the Sommel's point that a lot of universities, they do provide premium subscription, kind of a yeah, subsidization to Biorender's fees.
Okay, thank you.
Yeah. So should we move ahead? So everyone has access to their laptops, or who are joining from their email, mobile phones. So everyone is joining through laptops. So I hope you can do the things which I show here, and we can go. We will be on the same boat. So I hope. So let's go. So I will be sharing my screen. Let me know once you share. Uh, once you share, see my screen. So is my screen visible? Yes. No. Thank you.
Yes.
Great. So this is the Biorender Biorender main page. So where this is the website. So where you can see what are the features and all, and most of the, and since you talked about publications, University of Chicago, University of Cambridge, Roche, Hillcrest, University of Toronto, well, lots of universities use this. So there is no question of that the images are not publishable. So we can draw images of immunology, microbiology, neuroscience, and 30 plus fields. So I would request everyone to log in, to open this page. So did you open? Yes, no, कुछ तो बोलो. बिना बोले तो नहीं चलेगा मेरे वर्कशॉप में.
Yes, sir. We opened.
अरे आप ही बाकी लोग भी तो बोलो. 13-14 लोग हैं इस वर्कशॉप में. Yes, no, कुछ तो बोलो.
Yes.
Yes, we are following.
Yes, sir.
हाँ. So you have to sign in first. See, and we talked about that some educational institutes have provided Biorender. So for that you can click on sign up free, and then you can check whether using your institutional email ID. Suppose some people might be working in ISC, so they might have the name at the rate isc.ac.in. So with that email ID, they can check whether you have a free access to Biorender Premium or not. ऐसे तो since I am doing PhD in Bhabha Atomic Research Center, so I don't have access to Biorender. So here we will be using our normal. So you can click on continue with Google, or you can create an account. So if you want to create an account, you can click on sign up. It will again come work or institutional email ID. Instead, you can directly go and sign in using your Google account. So here I'll click sign in and continue with Google. So you can click. So already I have signed in. So this is the main dashboard of Biorender. So till now everyone is on the same page.
Yes.
Yeah,
So
sure.
page, yes or no, or any problems? So everyone is. Let me know. You can type in your chat box when everyone is on the same page. See, it's a hands-on workshop. So if you don't upload your screenshots, you won't be getting your certificates. Yes, let me know once everyone is on this same page. Then, if any problem, let me know. I'll be waiting for 2 to 3 minutes. Else moving forward. Yeah. Any problem? Are you all guys? So should I ask someone else to share the screen and show his laptop? So Kambati, can you share your screen? Are you on the same page?
One second.
Yeah, I'll stop my screen share. You can show that you're also working. Yeah, any one of you can just share your screen so that we can. Kambati, Niharika, you can share a screen instead. Yeah, great, cool. So we can move forward. Great. So this is the thing. So now we will click on Somalia's first illustrations. This already they have created a Canva page, the canvas where we can draw our items and all. So now, uh, so Dipta, what is your research interest?
So we are in cell biology.
सेल बायोलॉजी भैया बहुत बड़ा टॉपिक है सेल बायोलॉजी में क्या
Uh,
देख
we are now P38 and its effect on cell cycle.
P38 and
Its effects on the cell cycle in different cells.
okay, so you are a PhD student.
Yeah.
You are a PhD student or masters of
No,
bachelor?
no, no. I am second year's MSc.
Okay, great. सो अब आपने सेल बायोलॉजी बोल दिया है ठीक है वेरी गुड सेल बायोलॉजी अब शुरू कहाँ से होती है स्टार्ट्स फ्रॉम अ वेरी बेसिक सेल सो हेयर वी कैन सी देयर आर आइकन्स टेम्पलेट्स सब दिख रहा है ना यस या नो
Yes.
Yes. So here you can choose various templates are there that is popular in our field like cell biology, chemistry, or we can type simple cell. Like if you want, so I'll be using typing animal cell. So here you can see that something here it has come. Yes, yeah, no.
Yes.
Yes.
One second. Just a minute. Yeah. So it has come. ये क्या है? Animal cell है. So here you can take anything from your. So अब सब लोग ना अब since we have created our first image, ठीक है, using this. So अब इसमें image without labeling is called as very bad image in scientific. So how we can label it? So here there is option of text. Can you see? Yes or no? I guess.
Yes.
So oh, okay. So in the free version, we are limited for that 50 objects only we can get. So what we will do, we can group ungroup it and we can delete some of the objects. I will delete it, or rather we can take some simpler option. So like this, here we have to suppose we have to make this free. But the problem in it again it comes in the free version that only 50 objects they allow. So what are objects? So this all images are objects. So we will be creating something. So suppose this is a paracrine beta cell and suppose we have to start labeling it. So what we will do, we will take a line or arrows, we will do, and we can call it using, suppose we will call it nucleus. So you can adjust it. So or more refined, you can keep it tight and increase it. This is now better. Looks better. Cool. Yes, you know. So now what is this cell? So we can write here as Paradigm. So if you use it normally when we do in PowerPoint, now this kind of figure we don't get in PowerPoint. So we have to show a cell in form a circle. उसमें छोटा छोटा we that is the diagrammatic representation. But here we can show a actual cell which can be more legible and it will look more fascinating and it will look more eye-catching as well. So I hope now everyone should try their first image. Yeah,
Sir,
Raj?
yes, sir, I have a doubt.
Yes.
Sir, how can I add a second image in the same page? Or it is only one image will be added in one page?
Can add 50 objects in one image. We will be first everyone did
Sir,
their
there is first slide. I am trying to add in first slide. I add a structural overview of animal cell, a
so
label diagram of animal cell, and when I try to add
but
the diagram of beta cell, there it
that is
asks to take a premium.
yes, in free version in one slide we can add only 50 objects.
50 object means,
So whatever suppose this I have added, this is one object, this second object, this is third object, this is fourth object.
sorry, it is counting like one organelle as object or one complete cell as object.
One image, one organelle, one small thing. So suppose I'll copy that thing and show you.
Yes.
It is counting the objects. So search, applying template. So now this ungroup, this you see the squares are there now. The squares are they are counted as one object. So these icons are counted as one object. So in free version, we can only add up to 50 objects. So now I'll delete it. So now everyone try to create your any first figure, very simple figure. Everyone please try. I'll be asking to share your screen. So Raja, can you please share your screen? I'll stop my
Yes,
sharing.
sir, I'm at just adding my screen.
Yeah, or anyone who want to share. Masuma, can you try to share your screen and show what you have created in your first image?
Yes, sir, I'm trying to create.
Just
At
give me a
first,
minute.
I've created the one which you have created.
Okay, but try to show na. Try to show
Okay. Yes, yes.
and please do take screenshots so that you can upload in the feedback form so that we can check it that you have actually created and then you will be getting your certificates. So should I move forward? Yeah, Masuma. Yes, yeah, is yeah, great, very good. So you have used so now in Masuma's image, what all things we can add now? So first we can add a label, what kind of a cell it is. And since she has used a bend arrow, that is also very good. That is also a great thing. You can apply your creativity.
Sir, I have a doubt.
Okay, now, yeah, very good, Masuma. You can stop your screen share. Raja, you can start your screen share.
Sir, when I trying to add a arrow, it will ask for premium.
So can you share screen share? Then we can be on the same page. You
Yes,
can
sir, I
just
am just yeah.
so see. Yes, yes. Go ahead. Yeah, you can make it a full screen. Can you please of the screen
Yeah,
share?
yeah. My screen is visible, sir.
Yeah, make the biorender thing full screen.
Okay, done, sir. Now I'm trying to add a arrow, but it is asked for premium.
So the thing which you have taken, the cell which you have
I
taken,
wanted to label this glycoprotein. Yes, yes, sir.
glycoprotein, you want to be able, I can understand, but here the small objects are there. The small objects, they are counted as objects. So already the see you have, now you can add it. Now happened.
Somel, do you mind if I explain this?
Yeah, you can explain. You can go ahead.
Okay, so
Yes,
I'm
sir.
sharing
Now
my
it's work.
screen.
Sir, now it's working. Sir, now it's working.
Kirti, you want to say something? Please go ahead.
Yeah. I'm just sharing my screen. So, okay, Raja, the problem that you're having is,
Yes, ma'am.
see, you can see
Yes,
my
ma'am.
screen, right? Okay, so this is a diagram that I just made, right? So you see this
Yes,
blue
ma'am.
square outline.
Yes,
Do
ma'am.
you
Blue
see this
square.
blues? Yeah. So
Yes,
yeah,
ma'am.
this is
Yes,
one
ma'am.
object. Similarly, this is another object. Now these small five mannose molecule, these are five objects. So in total, how many objects do I have now? Five, six, seven, and this one mouse,
Six,
eight
seven,
object.
eight.
Now
Eight objects.
the non, yeah, the non premium version have just a limit of 40 or 50 objects. So if I were to add 42 more, then I won't be able to add any arrow, box, text, or anything else. So this is what Somwell is trying to convey you all. Okay. And the diagram that you just showed, it was, I think, a viral cell, right? So it had a
HIV.
circle inside a circle. Yeah, HIV structure. So it was a viral structure. So it has a double circle, then glycoproteins on the surface. So the glycoproteins were at least 8 or 10, then the circle is another object. So it already completed 50 objects. So you can't go any more than that.
Oh,
Got it?
yes,
Clear?
ma'am. Thank you, ma'am.
Okay. Somil, you can continue.
Am I audible now? Yeah. So here we will be creating an image where we will be showing in a cell a particular organelle is magnified. So we will use a simple cell. So I take suppose I take this, or now I want to show a piece of a piece of plasma membrane focused. So I'll add a bracket. So from here in this pic, what we can depict that we have taken a cell, and when we did a microscopy section, TEM microscopy section of a particular cell, we got to see that the plasma membrane is a lipid bilayer, double layered lipid structure where we can see the extracellular space and the cell membrane is there and intracellular space is there. So this type of images also we can create. Now we will be creating some enzyme. You might have seen in papers that some enzymes are there, some substrates are there, and some reactions also happens takes place. So we will choose some substrate. So this is a substrate. So this is an enzyme. So with the plus sign I can increase. Now when an enzyme, so we will reveal it later on. So when an enzyme binds to substrate, what happens? There is a formation of enzyme substrate complex. Enzyme substrate. So here we got one enzyme substrate complex. So since enzyme substrate complex comes in the transition phase, so here we can create, now we have to create a product. So we got to know that we have created some product. So it is a very basic thing. It is a very generic. It is not very specific to it. So you can create, you can now search for different, you can use PDB IDs. Suppose if I search for carbonic anhydrase. So you can add a structure of like structure of carbonic anhydrase and all using that PDB using PDB ID that we will be covering in part 2. So this we have created. Now we will be adding some labels to it. So this is substrate. Now this is our enzyme. This is enzyme substrate complex. And this is some product. So what we have tried to depict here. So we are showing here the lock and key mechanism of enzyme substrate reaction. So here we can give a title to our image where we see show that lock and key mechanism of enzyme substrate reaction. So I'll do it center bold. Up, I'm not liking this white color of thing. So what should I do? So now what will I do? Search. So meanwhile, I'm searching that option. You can try or and make your first image like this. You can, if you want, so it's a good image. So you can try to create your first image, and then we can share your screen. You can show on your sharing the screen. And meanwhile, I'll be exploring some more options. Yeah. Can you please go ahead, all of you, to go ahead and start your making your first proper image using all the labels and all. So should I stop my screen share? So now you have 5 to 7 minutes to make your first diagram, proper labeled diagram. Yes, start. Your time starts now. Now, if I want just a second, you can look here that if I want to change the color of the canvas, means my background, what I will do here, the size is there. I can choose the canvas color and use something good. So like this, it is more looking very more scientifically, more eye catching. No? Yes, yes, yeah, no.
Hi, hello.
Yes,
So can you
yes, yes.
repeat the process of changing the colors?
So when you see the in the canvas here, you can see there is a size also. You can see
Yeah, yeah.
now here it is a canvas color in a small circle organ. So what I'll do, I suppose I want to something else. It is not very good like this, I know. So you can choose any color whatever you like, or you can write the hex codes of the color from you can take it from Google and write the hex codes. So here it is the option for changing the canvas color. So here in this canvas, then canvas size, if you want to say change the canvas size, it is generally it is already 10 in 10 into 7, which is the fixed one. If you want to change, then you can change it also. You can change here, it got changed, but we will keep 10 is to 7 only. And done. Here from that you can use any color you can want. If you want to choose any color which you like but you don't know the hex code, you can click using a color picker also. So what I will do, so let me pick this color picker. Suppose this way it got changed to this, or again I use color picker if I want some this image or this image. This also you can create, but I'll take a since my so the background should be that how what type of things are there in my image. The background should be contrasting to it so that the background should not interfere with that. Yes. So I hope I'm clear and all. So now everyone might have created their first labeled image using many objects. So I would request anyone to share your screen first. I'll be stopping mine. So anyone voluntarily please share your screen so that we can see what you have created, and you have to explain that image a little bit. Thoda sa information.
Yes, sir, I'm sharing my screen. It is visible.
So what we have created.
Sir, I
So,
created this structure of HIV virus.
okay, but the correction is here. You are going, your diagram should remain in the canvas. So you can change the size of the canvas.
Canvas.
Yes, you can change the size. You can increase the size. Go to custom
Yes, sir.
aspect ratio. Six, you can click on 16:9. Oh, it out. Reset size. Click on reset size. Yeah, you can make the thing small, and then so that the images are in canvas, because when you will be exporting it, it won't be there in your image. So you can make it small. So you first make the size small and keep it in the white background. You can use 10:7.
10:7.
Yes. 10 is to you do it 10:7 and click on done and make the size small of your virus, and you can increase the size of your labels.
Virus.
हाँ, वायरस को छोटा कर दो, लेबल्स को छोटा
Okay.
कर दो,
And
हमारा इमेज है ना वो व्हाइट बॉक्स के अंदर ही रहे। अगर तो
okay.
हम व्हाइट बॉक्स बाहर लेके जाएंगे ना, तो अब जब हम एक्सपोर्ट करेंगे वो एक्सपोर्ट नहीं होगा फिर।
Yes, sir.
So now you can stop your screen share and edit this image.
Okay, sir. Thank you.
Anyone who is ready with their first image?
Yes, sir, I would like to share.
Yeah, you can go ahead. Yeah, what you have created. So first, before going ahead, you can give your small introduction.
Yes, sir. I'm an undergrad student of semester one. I've just started.
Yeah, where? From where semester one?
I'm doing BSc in biochemistry from Gujarat University.
Great. So how did you get to know about this work?
Actually, I found this on LinkedIn, and one of my professor also told me about this.
Oh, great, thank you. So now what you have created? Tell me.
This was the one which I've created, like the microscope here.
Yes.
So I've shown the eyepiece and the objective lens here. I did not get much time to do it, and
Yeah,
I've
you
created
so you can do other things and you can create a well-labeled image of this.
microscope.
Yeah, that you can do.
Hmm.
So what did you like? What since you're an undergrad student, I would like. So it was the first time you got to experience this kind of software. So how did you like creating your first biology means publishable image? How did you like?
So what exactly is your question?
So how did you like creating your own image first? First time you are using this software, na?
Yes, for the very first time. Actually, before 1 to 2 weeks, I was thinking like how, for example, in biochemistry we have a lot of mechanism. So I thought to understand those mechanism better, I can create some images. So I was just thinking, and I just came across this like notice about this workshop. So I thought it would be better for me since my subject is like that. We have so many mechanisms, so I can make use of it for my personal purpose also. For example, in studies also. And And secondly, I'm also a part of my editorial team of my college where I write articles. So there we have to, create our own image to show the mechanisms or something. So there also I can make use of this.
Very good. So you find this workshop fruitful for you.
Yes, very much.
Yeah. So the thing is, we are since it's a basic, a part one only. So after this, I will be showing how we can go to export and all. There are many how we will be creating pathways and all, and we will be importing from PDB IDs. We will do a series of this biorender workshop so that you can get experienced a lot. So yeah, you have created a very nice things. You can explore it more. So anyone else apart from us, you want to share your screen? Anisha, Abhijit, Anvita, anyone? Hmm. See, I'm not a professor that you were fearing from me and all. I'm a PhD student, so डरने की जरूरत नहीं. You can be informal. Yes, anyone else?
All of
अरे
us here are PhD students, so you can ask anything. You don't have to hesitate or shy.
हाँ, कुछ भी पूछ सकते हो कि PhD में एडमिशन कैसे लेना है और इंटरव्यूज कैसे देना है वो भी पूछ सकते हो. कुछ बनाया कि नहीं बनाया? भाई सर्टिफिकेट नहीं मिलेगा अगर नहीं बनाओगे तो. Say, you can. भूमिका, yes. अरे बिंदास बोलो, no one is going to judge you. Yes, भूमिका.
एक्चुअली मेरा ये वर्क नहीं कर रहा है इस पे। मैं फ़ोन पे वेबसाइट ओपन किया है ना।
अच्छा, अपने फोन पे आपने लैपटॉप पे नहीं किया है क्या?
नहीं।
हाँ, फोन पे थोड़ा इशू रहेगा बनाने में. एक तो स्क्रीन छोटी होती है फोन पे. आपने अपने लैप को आपने पूरा वर्कशॉप देखा ना अच्छे से?
हाँ हाँ।
आप अपने लैपटॉप पे ट्राई करना ना, फिर मैं फीडबैक फॉर्म आपको भी दे दूंगा. आप अपने पास उसको सेव करके रखना. फिर आप जब अपलोड करोगे इमेज ना, तो ही हम वो फीडबैक फॉर्म एक्सेप्ट कर पाएंगे. ठीक है, तभी हम आपको सर्टिफिकेट दे पाएंगे. आपको कोई ना कोई इमेज अपलोड करना ही पड़ेगा अपने फीडबैक
वेयर
फॉर्म में.
वी हैव टू अपलोड इट?
हाँ, वो मैं
कहाँ?
फीडबैक फॉर्म. खत्म कहाँ हुआ है? खत्म तो होने दो.
ओके।
यस. Anyone else, or should we move ahead? Now I will show how to export it on. Anyone else? You want to show your screen?
Can I show?
Same issue with me, sir.
आप भी फोन से चला रहे हो क्या, सुभिक्षा?
Yes, selfie. Yes.
अरे यार, फोन का जमाना छोड़ो, लैपटॉप पे आओ. लैपटॉप से करो ये सब थी. When you are doing science or you are creating an image, try to use your own laptop or PC. फोन पे वो अच्छे से एडिटिंग थोड़ी वीडियो छोटा छोटा दिखेगा, समझ में आधा समझ में आएगा तो समझ में नहीं आएगा. Anisha, you are telling something. You opened your mic.
Yeah, I just said कि same issue is with me because मेरे लैपटॉप पे issue है, so अभी मुझे उसको correct करना पड़ेगा. So currently I am using my phone.
Okay. So see, try to use for such things now. Try to use now. You can use your laptops.
Yeah,
So you
for earlier one, I am using the canvas for designing and all thing, and apart from that, I used Word itself to draw any type of image using the shapes and other tools which is there in the Word file. So this is new to me. It's good to know about the Biorender app and their features. So I will try this one. and that's it.
You said you already used Canva.
Yes, yes, मैंने
So
कैनवा यूज किया है। मैंने
what
एक और ऐप है लाइक समथिंग अदर वन, आई डिडंट गेट द name, so I also use that also.
what difference did you find?
Okay, so in Biorender there is auto prepared shapes which we don't have to create and edit, so it will be fast.
No, no, no. What difference did you find in Canva and Biorender?
Like there is many similarity between both, but the difference is there is ready-made shapes like for the enzyme purpose and for the viruses and cells, so we don't have to create that.
Yeah, that's a really great thing.
Yeah,
So you find it interesting?
yeah, it is.
Yeah. Raja, you raised your hand. You want to show something? Speak something? Yes,
Sir,
Raja.
yes, sir, I corrected my diagram. Can I share my
Yes.
screen again?
Go ahead.
Yes, yeah, thank you, sir. This is my diagram, sir,
Uh,
that I use.
going.
Sir,
You have to shorten these labels. The arrow marks which you have made.
yes, sir.
You shorten this to make it into the white space only.
The arrows.
Yes, yes,
Sir, is there any tool to select all of the elements together in a one go?
yes. I will show you. Yes,
Okay,
yes.
so he can use that tool na, so that it will be fast and shorten in one go.
That I don't know now how he has created the image and all.
Okay.
सर सर एरोस को सॉर्ट करने में फिर ये बहुत सारे कंपोनेंट्स हो रहे तो वो मार्क नहीं हो पा रहे अच्छे से।
हाँ, अब वो हमें स्ट्रेटेजिकली देखना पड़ेगा कि कैसे हमें मार्क करना है। तो हमें लगेगा कि वो इमेज छोटा है, लेकिन वो इमेज छोटा नहीं होता, वो अच्छे से ही मार्क होता है। तो वेट, लेट मी शो यू। वेट, अपना स्क्रीन शेयर बंद करो, मैं स्क्रीन शेयर करता हूँ।
ओके सर।
सपोज सबको छोटा करना है, एक साथ किया, ऐसे छोटा हो सकता है। देखो सबको एक, अब ऐसा मैंने कैसे किया? तो व्हाट आई विल डू, पहले मैंने इसको सिलेक्ट किया। नाउ आई प्रेस कंट्रोल ऑन योर की, क्लिक दिस, click this, click this, click this, click this। जो जो सिलेक्ट करना है सबको सिलेक्ट कर लो एंड सबको एक साथ छोटा कर दो। वो प्रोपोर्शन में अपने आप छोटा हो जाएगा। बट सिंस इट्स दिस थिंग, सो मैं स्क्रीन शेयर किया ही है। सो आई विल शो यू हाउ टू export this image so that you can use it in PowerPoint. So now what we will do, hub, sorry. Now we will be exporting it. So transparent background, it comes with the premium feature that we cannot use. So अगर हमारे पास प्रीमियम फीचर होता तो हम लोग ये अच्छा वाला रेजुलेशन लिए यूज कर सकते थे। बट since we don't have, so you have to use in this and you will do export. So now this is exported, that we can be using in PowerPoint. So I'll use a PowerPoint, copy PowerPoint, paste. Okay. अब इसको जहां तुम लगाना चाहो यूज कर सकते हो. Understood this much. Now अब हमें अच्छा इमेज चाहिए तो कैसे करें? So in a journal article, if you have created an image from Biorender, so you have to cite them. So how we will be citing it? We will paste. We will write this. So Biorender के image में no one will tell that there is any copyright or thing. समझ में आया सबको इतना? Yes या no?
So this has to be done every time when you're publishing an illustration citation.
Yes. No.
And also
Means
this
if
created in biorender.com watermark also has to be there.
so you have all the images you have created, so you have to, when you mention in the acknowledgement that you have created the images in biorender.com and you have to create it in this, you have to copy it and paste in your research article. This one line you have to write it, not in every image but in a collectively, that all the a labeled, all the schematic images whatever you have created, they are created in by https://biorender.com. So this is the citation of the how to we give citation in Biorender. And this hallmark only when we can remove only when we don't have, we have the premium access. Now if you don't want, you want a good thing later. If you want, so what I will do, I can this also you can, I can take a screenshot of it. So I'll take a screenshot of it. Simple. Now this is called chori karna. That I will make a image, take a screenshot of it. That also I can copy and paste. But you don't do it. That is not a ethical way of doing of using Biorender. So this you can use by using this Biorender. So there are now other free softwares as well available also, like illustrate.co.in, bio icons. So that we will be covering up in the next set of workshops, because time is also important and we have around 1 hour we have completed here, and we have made a successful one good image. So now I guess everyone got something to it. So to the audience, I would like to ask, what kind of expectations did you have from this workshop, as and was it fulfilled or not? Anyone? Else I have to take the names.
Yes, sir. All our expectations were fully filled. Like it was as it is, which I was expecting it to be, and I think much better than that.
Yeah, anyone?
Yes sir, it is very helpful. प्रोफेसर सर जब ऐसे डायग्राम्स यूज करते थे तो आई वाज वेरी वंडर्ड कि कैसे बनाते होंगे ये. अभी
Yes.
मुझे काफी इजी लग रहा है.
तो पार्ट वन था ये वर्कशॉप का. तो I will be now uploading the feedback form. So where you have to go through it and upload your first image. Then we will be investigating and we will be finding some, we will be scrutinizing it. If any corrections are there, I'll be letting you know. If corrections are not there, you will be getting your certificate for part one. And if corrections are there, you have to redo and upload the corrected one. Is it fine?
Yes, sir.
Yes, sir.
So free में तो certificate नहीं मिलने वाला. You have to work for it. So let me upload the feedback form. Just a second. And those who have done on mobile, you have time till today, 12 o'clock midnight, that you can use your laptop or some other's laptop to create your first image and upload it. And please do save this link in some or the other way. Please do save this link. Yeah, so if anyone has any doubts or questions regarding anything, let me know.
Sir, when we will have part 2 of this workshop?
Whenever you guys want, we will have. Simply, but we will have, since uh, you all, I guess you all liked it, so I'll be doing this workshop very early soon, as soon as possible. But now it is your responsibility to bring your friends to our community as well, and so that we can get more people should be benefited from this workshop. And since we don't charge any money, so you don't have to worry about it. But if it would be really helpful if you can recommend your friends also to join our workshop. And on every most of the Sundays we take interviews of scientists as well. You can go on our page, you can go on our web. So let me take a tour of our website as well, the SciComm Synapse. So here we do we do take interviews of scientists as well, that how they have become such a great scientist and all. So many great people like Professor Samrat Mukhopadhyay from ISR Mohali, then Dr. Amir Edkal, Dr. Lipi Thukral, then Vinay K. Nandi Kuri, who is the director of CSIR CMB, he also joined us. And you can go to it, you can visit our YouTube channel as well. So YouTube link is here only, where you can get all our live videos and all. So from that you can go to and see whatever videos, whatever you want to like. So it would be really helpful if you can recommend all your friends to join our community. And I guess my laptop has hanged up, so there is some issue. So Kirti, would you like to add something?
Uh, yeah, so what Sommel just showed you, it was uh, our weekly Sunday talks with scientists. Apart from that, this Kala Tattva is also one branch of what we do, where we showcase basically science communication in form of scientific illustrations. So just like Biorender, last to last Sunday we did a workshop as well with the famous science illustrator Dr. Ipsa Jain. And apart from Kala Tattva and scientific talks every weekend, we are also launching Ecosphere, which is a journal club. So in Ecosphere, we'll basically invite early career scientists and early postdoctoral researchers and even PhD students as well to talk about their research, and we'll scrutinize that research, and we'll discuss. It's like a consortium of discussion of their research and whatever input our audience can put in. So any stage of audience are welcome to join. You can be a BSc student, MSc student, PhD, whoever wants to join is very welcome. So we'll be launching that from next year onwards. So you guys can also join in that as well.
Yeah. So, anyone else who would like to add anything, or should we?
Any questions about SciComm or about Biorender or about any other thing that you guys have, you can ask.
Okay, so in the journal, do we have to contribute something, or do we have to just engage and discuss regarding the research work?
No, you don't have to contribute something if like monetarily or scientifically or in a presentation. No, we'll invite a speaker. They will present whatever they work on, and audience are most welcome to discuss, ask questions, get insights, informal discussion kind of stuff. Okay.
Okay. Okay.
How to upload image?
That will be discussed in part 2. Stay tuned.
Let me share you the feedback form as a screen share how you will be uploading your image. So this is the feedback form which I have shared you all. So this has opened. You will be writing your email, name, which workshop you, which university you are studying, and this it's your what is your feedback, any suggestion, and if you have any speaker suggestion, you will be definitely inviting them. Now please upload your Biorender workshop creativity. So maximum file we have, it is 10 MB. So you have to click on add file. So browse in a, if this is my first illustration, I will be clicking OK. Ah, okay, since this Gmail account, it is my full, it is completely 15 GB has been filled, so it is not uploading. But I'll be switching. I don't have any other account, so I'll be with. Let me open with my दूसरा account. Copy, paste. So here I can
Got it.
upload, browse, downloads. Okay. So it got uploaded. So when you will upload and submit it, it will automatically come to us. So we will scrutinize we will check your image. If no corrections are there, you will get your certificate. And if any corrections are there, we will tell you to make another image and fill up this feedback form again. That's why I said you save this feedback form link somewhere so that you can get access to it. So I guess we are done with the today's workshop. So I would be requesting Kirti that please can you give the formal vote of thanks.
Okay, so thank you everyone for joining in on Sunday. I know it's a weekend and it's a lot of effort for you guys and us as well to wake up, log in, and attend the workshop. And I appreciate that. The team of SciComm also appreciate that. So don't forget to fill your feedback forms, upload your images, and the part two will be uh, is we will like interacting with the part two very soon, wherein we will be discussing how to transfer the images or export the images in the publication and the PDB part as well. So stay tuned for that, and that's it. That's all. Thank you so much.
Yeah, in the part two also, we will be creating the images based on PDB IDs, how to use public data bank, protein data, public, not public, protein data bank, and how to use artificial intelligence also. That also we will be seeing in further workshops as well. So if now we can say goodbye and let's be connected, and we will be seeing you all in the next workshop. Yeah, thank you.
Thank you so much.